to Technically Speaking, where scientists and engineers come together to chat about a common interest, share knowledge, and satisfy some curiosity. I'm Ellie, and today I'm joined by Laura to talk about space moss and what it takes to survive in the deep cold of space. I came across this at work and I thought, why? Why are we doing this? Why are we taking moss to space? What is the benefit? But it is pretty cool. I've since learned, I've come around to the idea of space moss, and then basically I brought it to Laura and I said, let's talk about space moss. And Laura was like, but I let you fill in your blank.
That is usually how I respond to everything. Like, what do I think about this? I'll come back to you. I did do a lot of research on this. And I think you get to hear about some really cool science in your job because you work in a science news outlet. So you must get to hear a lot of fun stuff.
Yeah, that is true.
And my take on this, because I have a background in radiation science, was basically moss in space. What about radiation? How does it survive the UV and whatever else is out there?
I think that was my question as well, because I was falsely under the impression that they had taken it to space within a rocket, you know, like in the ISS or something along those lines with themselves, and they were interacting with it. But I was completely wrong. I'm not afraid to say. They were on the outside, which I think is potentially the coolest part of this.
I feel like that means they were kind of growing it on the side of a ship, like, you know, barnacles on a sailing ship.
Yeah, a bit of algae. A bit of moss.
But I think from the research paper that you shared with me, it was this like really highly engineered complex sample chamber that had different filters for different things. They had different samples to test different aspects of being in space.
Yes. I think we should probably wind it back a little bit to start with. They had the moss in a lab initially, and they did some like modelling and some testing of whether thought it would survive at different parameters. And then they decided after that, go on then, let's stick it on a big rocket ship.
Yeah, which seems sensible because I think getting an experiment on the International Space Station is probably quite highly competed and probably quite expensive. So you'd want to be sure of what you're testing before you go ahead and do it.
Yeah, I imagine they don't just let anyone stick a bit of weed on the side of their very expensive International Space Station. But yeah, it was cool. So basically, I think did they have 3 structures?
Yeah, I'm slightly confused by this, not being a plant biologist. I think of it as just different life cycle stages of the plant, but I don't know if that's particularly correct. But I'm going to go with it because it makes sense in my head.
I think you're not far off. I think they had 3 stages that they tested in the simulated space environment in the lab, which was pretty cool. And then they found that the moss spores encased in a structure known as a sporangium, fun word, were the most resilient. And then they sent those sporangium into space.
Yeah, and the sort of the simulated conditions. I'm mildly surprised they didn't try and simulate microgravity because I know from previous podcast episodes that you can do it in the lab. You can basically just keep rotating your sample so it never really knows which way is up.
Oh, wow. Okay, so like proper just confuse the heck out of it.
Yeah, but maybe they decided it wasn't a very good proxy for microgravity or wasn't really necessary. They didn't expect it to have much of an effect. So they just concentrated on, I think it was UVC and
yes.
pressures or vacuum and temperatures.
Yeah, they did UVC radiation levels that exceeded 100,000 joules per square metre.
It's a very helpful unit, that. I did try and convert it into a unit I'm more familiar with, which is the Gray. And I suspect my maths is a little bit dodgy. But I basically worked it out at 12 Gray per spore, which is quite high.
I don't know anything about Gray, but I'm willing to take your word for it.
So if you ever need radiation therapy to treat breast cancer, you receive, I think it's 4.4 Gray every day for 5 days. And your breast tissue is fine with that for the most part.
Wow, but obviously the like tumour site is not.
Yeah, that's correct, yeah. The tumour can't really survive the repeat irradiations, whereas the breast tissue can recover.
That's interesting.
Yeah. However, an entire body dose to a human of 5 Gray would kill about 50% of the population.
Hang on.
Yeah, that doesn't sound right, does it? It's about this more than your breast tissue can handle. Because of mucous membranes, they just don't respond well. They're really wet and stuff happens to them when they break down, whereas breast tissue doesn't really
doesn't have any mucous membrane.
I guess, yeah.
So if you had like mouth cancer, you would be struggling with the radiation dose.
I presume they would use a different radiation regime. I only know about breast cancer, so I'd have to look up anything else.
I was just trying to think of like a body part with loads of mucous membranes, and then you'd be struggling.
Yeah, but yeah, throat, digestive system, that sort of thing. But still, these seeds have survived quite a lot. They didn't really say what the dose rate was, and I guess because it's UV radiation rather than properly ionizing radiation, you don't necessarily need to talk about it in like, here's the dose and here's the dose rate.
Yeah, I guess it's also like constant exposure, right? Wasn't like we're going to zap them periodically with this. It was just like they're going to live in this environment that's got very high UVC the whole time.
Yeah, and I did start off thinking, well, isn't UV somewhat ionizing? Because it's pretty close to X-rays and gamma rays on the electromagnetic spectrum. But I don't necessarily think it is. It does still damage DNA, though, in a similar way that ionizing radiation does. It creates reactive chemicals, essentially, that then go on to react with DNA.
Yeah, you think then that the plants would be equally, if not worse, affected because they're much thinner. The leaves of like basic moss that you find in the woods surely is much less dense than like human tissue. So then you'd think it would really like mess it up.
Yeah, and I associate moss with surviving in quite damp conditions, like it needs that dampness to grow. But this is just the spores, isn't it? So they're different.
Yeah, this is like one structure that they use, and these were attached on the outside of the ISS in sample holders with different filter settings and left for 9 months. That is a lot of conditions to withstand, I suppose, for 9 months, because you've got loads of radiation, the fact that it's absolutely freezing, the fact that it's a vacuum, that probably is messing you up in some way, I guess.
What surprised me, not knowing much about plant biology, is that it was the visible light that seemed to do the most damage, not the UV. They didn't test for ionizing radiation at all, by the way, which I also find a little bit strange. Because you definitely hear about cosmic radiation, which is essentially really fast moving atomic nuclei causing problems to astronauts.
Right, okay.
I guess they figured that's not a parameter we can really test, so we just won't.
Yeah, I guess maybe they don't have like a way to replicate that. I've honestly no idea.
No, but this is like a starting point, isn't it? First steps, test the things that we can easily compare to.
Definitely. We've got all this stuff. Let's do the things that we can do. But the most amazing thing really is that even after they stuck it to the ISS for 9 months, they then took it back to Earth and it survived and it still germinates, which is mind-boggling. You just think it would die. How is it surviving?
Yeah, it's a lot of extreme conditions to tolerate, isn't it? For a long time, you said 9 months.
Yeah, like 250 something days.
And they mentioned something about the structure. Well, they mentioned the spore itself and then the structure that surrounds the spore, but they don't really understand why both those things would protect it.
Not just protect it, but then not damage significantly at all, right? To then be able to go home and then germinate.
But then I also wonder, like, see, I've exposed tomato seedlings to a ridiculous amount of ionizing radiation.
Sounds like a fun Saturday afternoon.
Yeah, and they survived for weeks just sitting on the windowsill afterwards. It was the cumulative effect of the radiation as the plant tries to respond to it and fix it and couldn't.
Oh, okay.
That was the
So
problem.
it's like a critical stage where it's too much and it can't overcome it.
Yeah, it just kind of says, no, my cells are too badly damaged. I can't repair them all. That's it. I'm just going to slowly wither away.
We've reached the point of no return.
Yeah, so I did wonder if when the moss germinated back on Earth, if it then just gave up after a few weeks or had some sort of mutation that made it, I don't know, a different colour, or it made it really stunted or something.
Yeah, that's interesting. I didn't consider that. I just assumed that it was fine because it could still germinate, but probably like something fundamentally had changed within it, or maybe it hadn't.
Yeah, I mean, you use, again, it's ionizing radiation rather than the UV that they're talking about in the research article. But you do use ionizing radiation to modify seeds to have certain traits. I'm sure I've read research about making rice drought resistant using radiation.
That does actually ring a vague bell that they like blast it with radiation for some reason.
And I always think, how? How do they know this? How do they know which specific genes they will affect by doing this?
Yeah, it does seem a bit of a like flamethrower effect of like, well, we'll just blast it and hope for the best, but I'm sure there's much more work that goes into it than that.
Probably. And then I think looking at the biology and all the different genes that could be expressed or mutated in some way or aren't is probably quite a challenge. You have to home in on that one particular thing you want to change.
Yeah, absolutely. They did say, which I should mention, is that they did germinate after rehydration and warming. So that clearly something had changed within them. They were probably Well, it sounds like they were freezing cold and very thirsty.
So that they almost needed like nursing back to a state where they could do something useful in that case.
Yeah.
You couldn't just take them as they are and then like depend on how quickly they warm them back up. Does it say?
No, it says the samples were returned to Earth. Researchers found all showed high rates of germination. Even those that had been fully exposed to UV radiation in space had an 86% germination rate. However, a type of chlorophyll in the space-exposed samples showed signs of degradation.
Yeah, and that was the degradation they attributed to visible light rather than UV. And they kind of speculated that the spores might contain some chemicals that help them tolerate radiation damage. So normally what radiation does is it creates these reactive oxygen species in cells. Cells usually contain water. Water's made of oxygen, hydrogen, right?
Yeah.
So then you get this reactive oxygen floating around in the cell. That reacts with your DNA, causes damage to the DNA. But if you have an antioxidant that can react with that oxygen before it gets to your DNA, it can protect you or your seed or spore or whatever it is you sent into space.
Yeah. This is why you should eat blueberries, folks, to minimise those free radicals.
Yeah, and I think in that respect, to create those reactive oxygen species, UV and ionizing radiation have similar effects.
Or like damage to the...
Yeah, I feel like I need to keep really clearly in my head that UV and ionizing radiation are different things.
But similar.
Yeah.
In my head, they work in a very similar way.
Yes, they're all electromagnetic radiation, just at different frequencies.
So now we get on to how. How could this possibly be a thing that we could take a small species of what I believe is a fairly common earth moss and send it to space and bring it home, rehydrate it, and it's still fine? Like, what must it have to make that possible?
Because you said they tested other species in the lab that weren't as resilient, didn't you? So there must be something more to this particular species of moss.
I don't know if they tested other species, but they tested other structures.
Ah, okay. And other things have been sent into space before. The good old tardigrades, or water bears, seem to be the most famous.
Oh, I love a tardigrade.
They do look quite cute for something that is a microscopic organism.
Yeah, and also it's weirdly cute, right? Because they're like lumpy, kind of looks like a sleeping bag with a hole in its face and tiny little claws. It's not giving cute on the outset, but for some reason they really capture people's imagination.
And I guess one reason is because they seem so tolerant to loads of ridiculous conditions. Apparently scientists have fired them out of a gun to see what sort of speeds they can withstand or impacts, I think it was.
That is like an Ig Nobel Prize waiting to happen, that is. I love that idea. But did they live?
Yes.
Oh,
Studies show that tardigrades could survive being fired at speeds of up to 900 metres per second, which is faster than a bullet fired from a typical handgun. There you go. BBC Future, I think that's in.
That's super impressive. But then they also survive space conditions. Did they put them on the outside of the ISS, or did they just sort of let them waft around?
It definitely mentions surviving space, though, I assume, because they can survive heat up to 150 degrees Celsius and have been frozen to round about absolute zero and have been desiccated and exposed to vacuum and all sorts. That makes sense that you would have stuck it in the vacuum of space.
Yeah, why not?
Apparently, the reason they can do this, it's unsurprisingly, when you get down to it, it's all to do with their genetics and their ability to enter a suspended animation-like state.
See, this was what I was wondering about with the moss. Was it just, like sometimes species go dormant to protect themselves? Was it just in like a hibernation sort of stage for 9 months in these extreme conditions, and then when it got back to Earth, it's warmed up, conditions improve, it's like, okay, yeah, we can germinate now?
I wondered that as well, because tardigrades, when you try and desiccate them or dry them out, they do something that sort of packs their organs away, and various genes have been identified that make some sort of disordered protein that somehow protects all the organs as they sort of shrink them down and pack them away, so that when they're dried out, the organs don't get damaged by the lack of water.
That's cool. Tardigrades are just cool. Maybe the moss has something similar. But then why would it have ever evolved that, right? Because this is, like I was saying, is just pretty normal moss. So it wouldn't have any benefit particularly. Like, why would it need to survive temperatures of minus 200?
Because you wouldn't really see it on Earth. I guess it's probably not really intentional. It's just like a weird side effect of needing to survive extreme conditions. Because moss is quite old in terms of it's been around on Earth for like millions of years. Isn't that right?
Yeah, I think it's one of those ones that's like, we've had moss before we had trees and sharks and stuff.
Yeah, and I think there are a lot of weird things in nature that you think, what possible need could you have for that particular evolutionary trait?
Yeah.
I sometimes think some of them are accidental. There isn't really an evolutionary need for it. It's just a lot of evolution happens almost by accident. You get a
Yeah.
random mutation that in a lot of cases is beneficial, so then you get a new species. But then some mutations are just
Neither one nor the other.
yeah.
That I always remember that being a thing of being like, it doesn't have to be beneficial, it just has to not be detrimental. So if you've not suddenly, but if you've evolved the ability to survive in really cold temperatures and that's not making your life harder, then you just keep that ability unless you need it, a different one.
Exactly. And when it comes to surviving radiation, the tardigrades can, when they sense there's DNA damage, they can then start upregulating genes that make repair mechanisms. So it's not that they don't,
Okay, that's handy.
yeah, it's not that they don't get damaged, it's just that they're really good at repairing themselves.
I'd like one of those. Why haven't we got that? That's what we
I
need.
think research is ongoing to see if it could be useful. Because obviously, if you're going to send humans into space, like into Mars, say, they're going to be in space for years. They're going to be experiencing a lot of ionizing radiation. It's not necessarily UV, although they will see that as well.
Yeah.
And there is talk of looking at how this constant low dose of ionizing radiation over years would affect people, because it's different to suddenly giving someone a massive dose of radiation for like cancer therapy, say.
Oh, I see what you mean, like a steady exposure for 10 years versus like one great big zap.
Yeah, and then seeing if there are ways that you can protect against that, which could be, I don't know, I'm not too sure how you do it, if you can like use a tardigrade gene and genetically engineer people or plants or whatever you want to send.
Oh, that's pretty sci-fi. Let's do
Yeah.
that. Start blending tardigrade genes with people. What does this teach us? Because ultimately, was this a beneficial experiment? All we've learned is that it can survive on the outside of the ISS, but it still needs to be rewarmed and rehydrated to do anything. And I don't feel like it's going to be a thing where we suddenly start taking buckets of moss to Mars, because I don't think you can eat this, or maybe you can and you'd be alright, but it's not very nutritious. Is this, maybe we should try something, like a potato, like the Martian, or something we could actually use.
Maybe it's a first step to genetically engineering something that is food that can survive.
Oh, good point.
Take all these different traits of all these organisms and kind of mash them together.
Create a super plant that can survive and reproduce and grow food in space.
Yeah, with some gene editing. I'm going to say stuff. I know it's CRISPR-Cas9, and I know what that looks like, but I don't know what the experiment is to make gene editing happen.
Just a lot of snipping, isn't it? Snipping and putting in.
Yeah.
I don't know. I imagine the equivalent of what sewing it back together is in CRISPR terms.
But then when you think about how do they know exactly where to cut and what they're doing, and what's the mechanism that works? This is where my mind goes every time someone says we did a thing. I'm like, oh, yeah, what do the molecules look like?
But they look like just the little strands of DNA, don't they? And then they zoom in further and further until you get the base pairs, and then you get your scissors out and start snipping molecules.
I'm not sure it's necessarily like a physical pair of scissors though. It's another molecule that comes in and does something to the DNA to then insert a strand, a section, a gene or whatever it is into that part of broken strand.
I like the scissors image.
It's a lot easier to understand.
I'd be actually very upset if that wasn't true.
And you're also not really convinced by sending random things into space to see what happens.
I mean, I like it as a principle, but in terms of usefulness, I don't know if it's that useful. Is it that useful to know that tardigrades can survive in space? It's cool, but has that brought us anything practical?
Oh, probably not yet, because from what I'd seen of the research I was looking at, this is all within the last 10, 15 years or so.
Yeah.
And it can take quite a long time for some scientific discoveries to be realised as a useful thing.
Yes.
But certainly looking at how they are so resilient could be useful for understanding how other species aren't necessarily resilient, I guess, in the face of climate change.
Yeah, that's what I was going to say. If it's extreme conditions that it can survive, maybe we can use that as a model to then predict what other species can do, maybe, or like you said, start taking genes out and putting them in stuff to make them more heat tolerant or cold tolerant or whatever they need to be.
Yeah, although weirdly, tardigrades don't seem to be tolerant to climate change anyway, potentially. They only survive hotter temperatures if they can start this desiccation response first.
Yeah, it's no good if everything goes dormant, is it? Because that's that's not going to help unless conditions change, which...
Then again, if things do go so terribly wrong that stuff goes dormant and then the planet cools down again.
Yeah, unless it's like Shell and BP that go dormant, I think you might be in trouble.
Sometimes I think it's always nice to do some science, almost just for the sake of doing science, just to know something.
Yeah, to be fair, like, we have gained knowledge. We know things we didn't know before.
Yeah, and you never know where that knowledge might lead you. So if you do all your experiments with a view that you want to fix a problem, you might never actually find your solution because you're looking in the wrong place.
Yeah, that's true. That is true.
I've interviewed quite a few people and I asked them, so how did this come about? Like, it was kind of by accident. We put our sample in the sample holder the wrong way around and measured this new property.
Yeah, and suddenly it worked.
Yeah.
Yeah, they do say that, things come when you least expect them, so maybe we should be just sort of trying new stuff for the sake of it and see what happens.
And there's always some radiation science or something else someone can do as a result of sending a thing into space.
What would you send into space to, see what happened to it?
Oh, I honestly don't know. I've never really thought about it.
Have we sent just human tissue? I know this is a bit gross, but, we stuck human tissue on the outside of the ISS for, a while. Just to see, see the effect of the radiation and the temperature on it.
You'd have to either dry it out, which would invalidate the experiment, because we're not dried out beings. Or you'd have to stick it in some sort of sample holder that would retain the moisture.
Say, for example, it was a limb. So we've, stuck an arm to the outside of the ISS.
Waving around.
Presumably it's going to degrade quite a lot. In which case, whatever it's in is not going to be, if it's in a holder, it's going to be shrinking and then it's just going to float into space, in which case we've lost the data, which would be a great shame.
I think there are probably easier ways to test what would happen to an arm if you put it in a cold vacuum. You could do that on Earth.
Yeah, but it's not as fun, Laura.
Yeah, and I think the question for anyone doing a research proposal to send something into space is to, what will you learn that you can't learn here on Earth?
What's a really hardy houseplant that's really hard to kill? Maybe we should send one of them up, see how that survives.
Peace lilies seem to be pretty good. I've got one that I've nearly killed several times and it's always bounced back. Mint as well.
Mint?
I nearly killed that quite a few times.
Oh, this umbrella plant, actually. That's pretty hardy. That's survived a long time.
There you go. But then again, although my house is quite cold and dark at the minute, it's still not the vacuum of space.
Yeah, the temperature extremes are not quite at the level of what they would be outside the ISS. Well, I think that might have to be where we leave it before we start sending all sorts of things up there. We've covered space moss, and oh boy, is it exciting. It can survive all sorts of extreme temperatures, vacuums, radiation of all kinds, and still make it back to Earth in time to germinate. We've talked about how tardigrades can survive and just what they're made of, and also then what we would send to withstand the cold, harsh conditions outside the ISS. So if you enjoyed this episode, you'll have to go back into our back catalogue, because I'm afraid to say that this is the last one of the year, but there's plenty more for you to listen to before we see you in 2026.
Yes, we are taking a break over Christmas and we hope you enjoy the festive period, whether you're celebrating Christmas or not.
Yeah, we will see you very soon.
The views expressed in this podcast belong entirely to the person that said them. They do not represent any industry or organisation. If you enjoyed listening to these views, it would really help us out if you could rate us, leave a review and tell a friend. This podcast was sponsored by no one, but if you're interested in funding us to continue to have frank discussions about science and engineering, please get in touch.